Okay, the next entity that we'll be discussing is called a neurothekeoma, and this is the cellular variant of this entity. This is a curious entity in neural proliferations because the histogenesis of the cell is really unclear presently. And there's a broad spectrum of stromal changes that you can see that can make the histologic low-power appearance of this lesion quite variable. You can see anywhere from a hypocellular myxoid stroma all the way through to hypercellular presentation with a very densely pink collagenous stroma. This lesion really, I think, borders more on that hypercellular kind of collagenous stroma appearance. This lesion presents more commonly in young adults, and it can involve pretty much any site, but the head and neck region tends to be preferentially involved. Presents as firm pink-red to red-brown papules or nodules. It's a benign entity. It's usually asymptomatic, but occasionally tenderness has been reported with this entity. And histologically, you'll usually see a shaved biopsy because it's a dome-shaped papule, so you'll see a shaved sample. And you'll see these nests and clusters of cells or theques of cells. And the cells are usually spindled or epithelioid or both. So you can see distinct difference in the cell morphology in comparison to most of the other neural proliferations we've been looking at. These cells are a lot plumper. They have more abundant cytoplasm. I mentioned that the matrix is this densely pink collagenous matrix in this example. But you can see how the cells are organized into these small nests that are somewhat interconnecting. Very frequently, this tumor will have kind of an infiltrative quality to it in the manner with which these nests kind of dissect between the collagen bundles. It gives it sort of an infiltrative appearance. It tends to occupy most of the dermis and occasionally can extend into the subcutis as well. It tends to be ill-defined, so it's not a very well circumscribed proliferation typically. And when you look on higher power at the cells have these large kind of ovoid nuclei with ample eosinophilic cytoplasm, as you can see. And not uncommon to see some mitotic activity as well. We've seen a few mitoses already. So you can see these large ovoid nuclei, ample eosinophilic cytoplasm, very epithelioid appearing, not the wavy nuclei we're used to seeing in some of the other neural proliferations that we've been chatting about. Now, in terms of the staining pattern, this tumor is typically positive for NKI-C3, for PGP9, for MITF, and for neuron-specific enolase. It can be S100 positive, but S100 positivity tends to be quite focal and weak. So that's something to pay attention to, is that S100 may not be the most reliable marker for this neoplasm. So this is a neurothekeoma, and this is the cellular variant of a neurothekeoma.